So today we're going to be talking about a mineral that I think is often overlooked, but it's actually one of the first minerals that we look to correct in our consulting work with farmers, and that's manganese. And so today we'll be going over exactly what manganese does in our plants, how it functions in our soil, the importance of it in terms of plant production and disease management. We're talking about the fertilizers we can use to overcome deficiencies, and also how to manage manganese in a regenerative way so that we're not reliant on fertilizers into the future. So to begin, what is manganese? Manganese is one of the essential minerals for our plants, and there's a lot of really important functions that it plays. Typically what it does is it's required in a lot of enzymes as a cofactor or an activator of those enzymes, which means without manganese you don't actually get the function of those enzymes, either because you don't have the key component of it that actually allows the enzyme to do its thing, or you don't have the starter engine to start that enzyme, which is the activator. The most important enzyme that manganese is used for is what's called the oxygen evolution complex, also called the distorted chair, which you'll see here, is a very important enzyme used in photosynthesis for the splitting and breaking apart of water, called water hydrolysis. And so this step is super important for photosynthesis, and it's one of the 4 nutrients we're looking for managing photosynthesis. So that's nitrogen because of the ring around the chlorophyll, magnesium, iron, and manganese. Super important. So this is just as important for photosynthesis as all those other minerals. The reason why is that plants take up water, but they need to split that water into hydrogen, oxygen, and the electrons, which are then used in the electron transport chain. What manganese is very useful for is holding and passing on electrons. And so similar to iron, and we talked about that in our previous video, is that manganese, similar to iron, has multiple oxidation stages or phases. So manganese typically in the plant exists in the reduced form, or the form that has the most amount of electrons, and so has the lowest charge, of manganese 2+. There's 2 other phases in the plant, that is manganese 3+ and manganese 4+. These, although they can be in the plant, they're not that useful, and typically it exists in the 2+. What manganese is very useful for is that it can go undergo oxidative transformations to then lose and gain electrons, and that's super important, and we'll see why. So typically this contains a lot more electrons. Manganese 3+, you have the manganese 3+ plus an electron if you're going to split it or take it from manganese 2+, and then manganese 4+, if it was to be produced from manganese 2+, you'd get 2 extra electrons. So this is a very important function of manganese, and it's why it's used in a lot of these roles. But effectively in the water hydrolysis stage, the water gets split, and what happens here is that the manganese 3+ gets charged up with an extra electron from this system. So it goes from 3+ to 2+. Over here it spits out those spare electrons, which can be used, basically those electrons get energized. Once the light comes in on the pigment that receives the 680 nanometer wavelength, that goes off and does a thing, and so that electron is super important. The manganese 2+ gets stepped down to manganese 3+ to then be reused. The oxygen is split into hydrogen, which is then used for the proton pumps, and oxygen, which is then used in respiration. So overall, this is a very important step for photosynthesis, and it doesn't occur without it. And so you'll see here a graph of the oxygen evolving complex and its production in relation to manganese availability. So super important. It's also important to know that this doesn't quite have an effect on chlorophyll production. It's more so in this system. And my understanding is that under a manganese deficiency, you can actually get a reduction of the photosynthetic ability of the plant by 50%. So super important. You also find with this that I think the water efficiency should improve, but the most important factor is the increase in carbohydrates. So the water hydrolysis and the oxygen evolving complex is required for photosynthesis and therefore carbohydrate production. Everything else really stems from this, even a lot of the other benefits from manganese. Now, because manganese also is very effective as a oxidizing or reduction agent, it's used in what's called the manganese superoxide dismutase. Now, this enzyme effectively can take in electrons and store it there like a battery. That's really important for radical oxygen species, which are just a typical byproduct of photosynthesis, as well as any oxidative stress. So this is an antioxidant, which reduces the plants to a lot of stress factors and can allow it to function in a productive way. So an example of this is when the plant gets oversaturated with light, photosynthesis speeds up and produces all these radical oxygen species. You need manganese to produce the manganese superoxide dismutase so they can handle that buildup of free radical oxygen species. Now, this enzyme also has a very important function in plant defense, not so much in plant defense itself, but when a plant gets attacked by a pathogen, plant can do what's called a oxidative burst, and plant releases all these reactive oxygen species with the aim to damage the pathogen. So this here is used to soak up a lot of those oxidative agents so that the plant doesn't damage its own cells. So that's quite important for that. Another important enzyme that manganese is used for is in oxalic oxidase. And so this enzyme has two really important roles. The first one is it inhibits the toxins produced by fungal pathogens, quite important for increasing the tolerance of plants to fungal pathogens, as well as lignin production. So lignin production as well helps with creating a defense barrier for the plant to pathogens. So this contributes to plant defense against both fungal pathogens as well as just pathogens in general, because if the pathogens can't puncture through the cell wall and can't get into the plant. So lignin is quite important for that. Another very important process that manganese is used for is in the shikimate pathway. So this pathway, super important for developing plant secondary plant metabolites, which contribute to a lot of the quality components of the plant. So think of all the flavonoids, as well as lignin, as well as aromatic amino acids. So this is super important for developing kind of that flavor and aroma of our plants. Interestingly enough, the shikimate pathway is the pathway that actually gets shut down by Roundup or glyphosate, and so shutting this down is actually what kills the plant when you're using Roundup. And we'll talk about Roundup and manganese just in a moment, because there's a very interesting link or effect on each other. But so one of the byproducts of a manganese deficient plant is your products might taste dull or don't get that aromatic or flavorful component, which is, I think, super important when we're talking about quality of our products, and especially when people have the anecdotal experience of, they grow their tomatoes at home and it actually tastes like a tomato. What I'd suspect is that because of the soil health, and we'll talk about that in a moment, you get an increase in manganese, which then contributes to increased secondary plant metabolites, as well as a whole other bunch of functions, which is more related to the plant health pyramid and going down to lipid production. But overall, you need manganese for, or both your increase in photosynthetic ability, but also the shikimate pathway, which produces these secondary plant metabolites. Leading on from that, lipid production is quite important for lipid production in the sense of it firstly increases the carbohydrates, then be used in lipid production, but also in the production of fatty acids. And so all of that's required in lipid production. And finally, manganese is important for potassium regulation. Now, my understanding with this is that manganese is needed for the proton pumps with this process, which then regulates the uptake of potassium as well as many other cations. And so typically, if you're struggling to get potassium into the plant, but you actually have a lot of potassium in your soil, it might be a manganese deficiency in the plant. And we'll talk about the difference between a manganese deficiency in the plant and in the soil, because they're two completely different things. And there's a lot going on when considering getting manganese into the plant. But if you're having problems with potassium in the plant, either too much or too little, it might be a manganese problem. And so I mentioned that one of the most important functions of manganese is in the production of carbohydrates, and really everything flows on from that. So you'll see in this study here, they looked at manganese deficient plants and sufficient plants, and what they found is that you basically get a 4x increase in the soluble carbohydrates in the leaves, a doubling in the stem, and a massive improvement in the roots. So super important we have manganese for that. That flows on to the protein synthesis pathway. And so we need carbohydrates in order to convert the ammonium into amino acids. And so it's super important that we have these carbohydrates to do that. And you can see that as well in that table where the soluble N is really reduced when you have sufficient amounts of manganese because you got those carbohydrates. And so it's interesting also in that table that the protein levels don't really change. So I'm not sure what's happening there, and you get a slight increase in the biomass. And so just touching on the amount of carbohydrates in the roots. So that led to a 4x increase in the root size in the plants that had sufficient amounts of manganese. So overall, really important that we have manganese to increase the carbohydrates, to then increase everything else in the plant, especially the overall health of the plant. So how do we actually manage this in our soil? So in our soil, there's really two components. There's our total manganese, which is everything from what is available, but also all the unavailable components of manganese, either in the primary minerals or the secondary minerals in all their unavailable forms. That makes up our total manganese. Now, manganese is the 10th most abundant mineral in our soil. So typically, you're never actually that deficient in manganese in terms of our total component. Now, I have seen soils that are deficient, and we'll talk about what to do in that case. But typically, like iron, we're never actually truly deficient of manganese in the soil. Typically, it's a availability problem. And so manganese is only available in the manganese 2+ form. What this means is that under oxidizing conditions, manganese 2+ will go to manganese 3+ and 4+, which is not available to the plant, and the plant can't use it. So even on our soil tests, I'm not sure if the soil test in terms of availability actually picks up on these oxidized forms, or if it just considers manganese 2+. So what could happen is that you might have available manganese on your soil test, but it's actually not translating into available manganese on your SAP tests. One of the key things with this is that manganese 2+ could go to manganese 4+ or 3+ in a matter of hours, and likewise the opposite occurs. And so this is because the redox potential of your soil can change rapidly within the day, whether or not that's due to a herbicide application, which is highly oxidizing, or it could be get a rain event and it reduces the soil environment, meaning everything goes this way. It changes quite rapidly, which is a bit different to pH. pH changes quite slowly. Redox changes very, very rapidly. As part of our management strategy for manganese is that we want to make sure we have a reduced state for manganese. And we'll talk about that in a moment when talking about the regenerative management practices. But typically, you're going to get a shift to manganese 4+ when it's oxidized. So these are things like sandy soils, which are well oxygenated when you're using, say, a lot of nitrate fertilizers. You also get a reduction in available manganese when it's alkaline. On the other side of that, when it's reduced and acidic, you get a lot more manganese 2+. So this can be a problem, especially when you get very low pHs. You get too much manganese and it causes a toxicity, which is problematic on all sorts of reasons. It antagonizes uptake of all your other trace minerals, as well as calcium, magnesium, and then it can also cause a lot of oxidative stress on the plant. So too much of those radical oxygen species. Now, how do you know if you actually have enough total manganese and enough available manganese? Well, what we do with all the farmers we work with is at the start of our program, we take a total soil test. This soil test will tell you everything you need to know about your soil, and you only need to take it once because your totals don't really change that much. It's really more to do with your parent material, but it's important to know, do we actually have manganese in the total component? Because sometimes we don't. If you get a really sandy soil, that might not even have total manganese. So what we're looking for at a minimum in our total is about 200 parts per million. So if you have any less than that, what that means is that your soil, the parent material of your soil, just doesn't really have that much manganese, which means it might be beneficial to actually make a manganese application to your soil to boost your soil stocks of manganese. So this is quite rare. Typically don't get it, but sometimes you might see it. The next component is available manganese. Now, I'm not sure if it actually considers all three of these. Doesn't necessarily matter because it's going to swing either way within a few moments each day. Typically what we're looking at for this one is somewhere between 15 in a very sandy soil to about 25 in a heavy clay, and that's in parts per million. So what we're looking for is, firstly, do we have enough manganese in our total? If that's a yes, then we look at the available and we go, is the total manganese actually moving into what could be available? And let's just assume it's all of these three. That is likely due to just biological activity, releasing that manganese. We'll talk about getting in 432 plus in a moment, but typically we just really need it to hit that 15 to 25 parts per million. So 15 in sandy soil, 25 in more of that clay. If we're using the Haney soil test, which is what we like to use in future years after getting the total done, we're looking about that midpoint, so about 20 parts per million. So my understanding is this doesn't necessarily tell us whether or not it's in the 2+. And so what we do from here is use differential sap test analysis to know what the plant's actually picking up. So the differential sap test analysis is we take a leaf from the youngest leaf, the youngest fully developed leaf, and a sample from our oldest leaf. We'll send that off to the labs, and that will tell us how the plant is translocating those minerals. So manganese comes up through the roots and it gets deposited into the developing leaf. But what happens is, say the plant's developing like that and it puts out its first leaf, it gets loaded up with manganese. And then the plant continues its growing, it puts out its second leaf. But say during that time, the soil starts to dry out and becomes oxidized, which means the manganese 2+ moves into 3 and 4, and the plant can't access that manganese. It means that we're going to have less manganese in that leaf. So if we take, say, that leaf and we compare them, we can actually see, is the current soil environment giving the plant the more or less manganese as before? And so that's what we use the differential sap test analysis for. And it's important, you need all three parts. You need the total, you need the available, and the differential sap test analysis to know, is it in the plant? Some people say you just need this, or some people say you just need your soil test, but really it's a combination of all three, because that will inform you what part of your system is actually the bottleneck in supplying manganese. So if that's the case, then we do a foliar application. So if we're not getting enough manganese in our plant, in terms of we have enough soil, enough manganese over here, we're just not getting in the plant, that would warrant a foliar application. If we're not getting enough manganese because it's lacks in our total, potentially a soil application. This here is more likely a biological problem if we have enough in our total. And so what warrants a deficiency? Typically it's a 10% variation. That's the threshold. We don't tend to use threshold numbers in our differential sap test analysis just because there's not that much data. And so the ideal level of manganese in, say, our older leaves or our younger leaves will change depending on the plant, the stage it's up to in its development, as well as the plant species and variety. So to go, exactly 4 parts per million of manganese in the leaf is the ideal level. I wouldn't say it's quite accurate. It's a good idea to have something to aim for, but what I like to use is that 10% as that starting threshold to go, okay, it's starting to have problems either with uptake or a deficiencies coming on. And so I tend to like to use that. The problem with this is that if you did have an excess here and then the plant balances out to the ideal level, this would still suggest a deficiency. So there are problems with doing it this way, but I think it's just as valid as using threshold. And then you don't have to worry about, are you getting the right threshold numbers for both the plant species as well as the variety as well as the plant stage. So if you are using differential sap test analysis, we have a free analyzer that you can use on our website. You put your values into the analyzer tool and it sends you a report back saying whether or not you have that 10% variation and how severe that deficiency is. Again, it works across all varieties, all growth stages, all plants. It doesn't matter because we're just looking at that variation. We're looking at what is the plant actually telling us. Likewise, if you have a soil test you want analysed, you can put it into our free soil test analyzer. Get a nice report, some recommendations at the bottom, which are a rule of thumb. It's not official recommendation, it's just something you might want to compare to. That's all free on our website, so you can go check that out. Now, when it comes to actually looking at manganese deficiency in the field, what we're looking for is that youngest leaf. Now remember, it's immobile, which means it's going to get stuck in the older leaves. So we're looking at the youngest leaf, and what we're looking for is a yellowing of the plant parts further away from the veins. Similar to a magnesium deficiency, except that comes up in the oldest leaf first. A manganese deficiency comes up in the youngest because it's immobile, which means at any point in time, if the tap shuts off from our soil, feeding our plant with manganese, it'll cause a deficiency in the youngest leaf first. And so typically when we start working with farmers, and even throughout the years, we're looking at managing manganese pretty much as one of the first minerals we're looking to manage. That as well as magnesium, because this is super important for the photosynthetic ability of the plant, which drives everything else. So what we've been seeing this season is just so much ammonium in the plant that it's antagonizing a lot of other things. And so the only way to convert ammonium into complete proteins is with carbohydrates. So you need that carbohydrate backbone to attach that the ammonium onto for it to then be converted into the amino acids. So if you don't have the carbohydrates because you don't have manganese or iron or magnesium, then you simply get a buildup of ammonium, which is then going to attract a lot of pest and disease. So super important you manage this pretty much first. There's also the benefit of regulating your potassium. So if you have too much potassium, that's going to antagonize your calcium. If you antagonize your calcium, you're going to lead to a weaker plant. It's also going to hinder a lot of those critical points of influence. So if you want to check out critical points of influence, you can go check out the video, Critical Points of Influence. If you want to learn more about the metabolism of ammonium, you can go check that out with our regenerative nitrogen management video. When it comes to managing pest and disease with manganese, the most important components is the production of the lignin, as well as some of these compounds that inhibit fungal toxins. The other component is the secondary plant metabolites, which can also have anti-pathogen effects. Quite important. So manganese has been pretty well studied in terms of take-all in wheat. And if you have a copy of this book, it's a fantastic book. It will tell you basically everything you need to know about each of the minerals and how they relate to diseases. And so you can see here, there's a whole bunch of different tables and references to different effects of manganese on that particular disease in a particular plant. Take-all in wheat has been pretty well studied. What they conclude is that the main driver of this resistance is the increase in lignin in the roots. So that probably is due to the increased carbohydrate levels. And remember back to the table where there was a lot more soluble carbohydrates in the roots, and that also probably increases the root exudations, which then feed beneficial microbes in the root zone to then get rid of those pathogens. So there's that component, but also it increases lignin production because you need that carbohydrate backbone to then increase lignin. So it's one of those things where everything kind of plays together, but it all stems back to having sufficient amount of manganese. So a very important component of this is making sure you have sufficient amounts of manganese, and typically what drives a deficiency of manganese is other practices that we do which convert manganese from this reduced state into an oxidized state. So what are the things that we do that reduce manganese? And they are really any practice that causes oxidation in the soil. And so there's a very clear link between the effect of manganese availability, the practices we use, and take-all in wheat. And that's been very well studied. And you can see the table here on the practices, the effect of take-all, and manganese availability. So here are the key things that a lot of farmers do that decrease manganese availability. And so if this is the case, it's driving manganese from that available component to more of this 3 or 4, which is not available to the plant. So on your soil test, it might look fine, but you're actually getting a deficiency over this side because it's not available to the plant. Cool, so the first one is liming. So liming, although it's great and it increases the availability of a lot of your other minerals because you get that increase in pH to more that ideal, liming oxidizes the soil environment and it does reduce manganese. So you get both the hit of increasing or reducing acidity as well as oxidizing the soil environment. So this one, the way to manage against this, typically it's best to lime, right? We need to increase our pH oftentimes, and it is economical to do so. If that's the case, smaller amounts are better, and there is a fine line between too little, so say below 1 tonne per hectare of lime, typically is below the economic threshold for liming. So you're not going to get an economic response, or the cost of applying it outweighs the benefit you get. So typically it needs to be greater than 1 tonne per hectare. On the other side of that, if you're applying more than 4 tonnes a hectare, it's going to really antagonize everything else from a calcium point of view. Somewhere between that, you're not going to get as much of a reduction in manganese. So probably can't give you an exact number, but the more lime you apply, the greater the chance of reducing your manganese. Now this also is beneficial if you have toxic levels of manganese. Liming will help to reduce the toxicity of the manganese. Say you got a pH of 4.5, and you're getting manganese toxicity, liming will help with that, bring it up to a bit more of that acceptable range. The other component is using nitrate fertilizers. Nitrate oxidizes the soil environment. Likewise, probably with urea, which although it gets converted into ammonium, it then gets converted into nitrate. So I think net, it probably has an oxidizing effect. The next one is tillage. Tillage loosens up the soil environment and it oxygenates the soil, which also oxidizes the soil. You're either tilling or you're applying herbicides, which are also oxidizing. And so it's probably much for muchness. It really depends on what you want to do. I mean, it's easy to say, yeah, we've got to reduce our tillage or we've got to reduce our herbicide practices, but it's very difficult to actually do that. So don't kick yourself too hard if you're doing these things, because operationally you almost have to. It's just important to know that this will reduce our manganese availability. And so potentially, if we are using a lot of tillage, we just have to accept that we might have to increase the manganese applications with, say, a foliar. So the other thing I mentioned is a herbicide, particularly glyphosate. Glyphosate is a very effective chelator of manganese. So what happens is it oxidizes the manganese to an oxidized form, so 3 or 4, and it chelates it and makes it unavailable for the plant to use both in the soil, but also in the plant of resistant plants. So typically that's why we're probably seeing a lot of manganese deficiency despite having so much in our soil. And it's typically because we're either tilling up our soil or applying a lot of herbicides, which are going to oxidize the manganese to an unavailable form. Now there's some interesting links with GMO or the Roundup Ready. There's typically a problem for those plants to actually pick up manganese in the first place. My understanding is that those plants tend to oxidize the soil environment, which reduces manganese availability. And so there's typically problems with that. Likewise, if you're then applying Roundup or glyphosate over the top of that, you're going to then continue to reduce your manganese availability. Another one is bare ground. Bare ground will also oxidize the soil environment as the soil is exposed to light. And then finally, a really interesting one, which you probably wouldn't expect, is actually using manure. So manuring will oxidize or can oxidize the soil environment. So sometimes we've got a few clients that are using local sources of manures, so pig manures and chicken manures. Although they're great for phosphorus, they can oxidize the soil environment and reduce our manganese uptake. So all these things, there's benefits of all these things. Like using lime, there's benefits to increase your pH, increase the availability of other minerals. Nitrate, using more pathways for nitrogen uptake for your plant can be beneficial. Tillage, you're not using a herbicide, you can do a light disking at the top, might be beneficial for controlling weeds without using a herbicide. Herbicides, quick and easy, whatever. There's a lot of benefits to a lot of these things, so I'm not saying don't use them, just know that if you are using them, you're going to be reducing your manganese availability. So the question is, despite doing these things, what can we do to also help with our manganese uptake, because it's so important for all these things? And that's where our regenerative management strategies come into place. So the first one is getting a good soil test and making sure you actually have manganese in your soil. So in the total component and the available component. Say you don't have enough manganese in your total soil environment, it means that you then need to apply some manganese. Now typically the best form to soil apply manganese with is manganese sulfate. So a soil application of manganese sulfate. The problem with soil applications is that it can rapidly convert into unavailable manganese, which is not necessarily a bad thing if we don't have total manganese in the first place, because we're just trying to build up the reserve. What we then need to be doing is looking at this level and going, okay, how do we push manganese over into this available form? This push is typically later in the in our regenerative management process. What we'll then probably be looking at is a foliar application. So we then take our differential sap tests. Now, if we're deficient in manganese, and typically we are, what we'll do is either another application of manganese sulfate as a foliar. With that though, you want to make sure you apply fulvic acid. So fulvic acid will help to chelate this, help with the uptake of the plant, and the other important thing is that it keeps it in the reduced state for longer. What can happen is if you apply this onto a leaf and it dries out, it can then oxidize on the leaf surface. So you're not really getting all the benefit. So we want to make sure we are applying fulvic acid with our application, especially for our foliars. That will help the uptake. Now we can use manganese sulfate, or you can use a chelated manganese. I'm not sure if there's much of a difference. If you're going to use manganese sulfate, make sure to add a fulvic acid to it, or some other chelating agents. There's a few amino acids out there. I think fulvic acid's super good enough, and I think it might be the cheapest too. But that's up to you. You can buy from some suppliers that already have that fulvic acid in there. Likewise, you can get it from other suppliers which use a different chelating agent. I'd probably go with the fulvic acid or some kind of amino acid, just because it's an organic component, which means it contains carbon, so the plant can then use those carbons for later. What these two things allow us to do is maintain our production and increase our manganese levels in our plants so that we're not losing production. This is super important, and it's great to get all ideological about, we shouldn't be needing to use herbicides or whatever. But the thing is, if there's a few changes we can do in our system and it gets a big uplift in our photosynthetic ability of our plant to then improve the soil, I'd much rather do that than go on this ideological journey of, hey, let's just cut out our fertiliser and hope that the system works. That doesn't happen. So after our soil test, after our sap test and fixing all these things, the plant will go into a higher level of photosynthetic ability, producing more carbohydrates to then increase yield and also improve the soil environment. So that's a big component on how we're actually going to transition from this stage of applying nutrition to the soil's going to apply it for us. So this is going to increase plant health. That is going to then increase soil aggregation. So the whole aim of our regenerative management is we need to increase soil aggregation. And so soil aggregation is super important because it allows for these little pockets of reduced soil environment. So if you think of the soil environment either as super sandy and super loose, like this, that's going to be well oxygenated, say after ploughing. What we want is to be able to convert this into zones of good aggregation. What that allows is good oxygenation or good aeration in this part in between our aggregates, but also got these zones of a reduced soil environment, which is then full of available manganese. Our roots can explore deep into the soil profile for water and more nutrients, but also go into these aggregates for the correct oxidation stage of our minerals. So that is a big component of managing our soil in a regenerative way. What that means is we need to do things that increase aggregation. So the first one is increasing our plant health, especially in areas where we can't have a cover crop because of water limitations. Increasing the your plant health of the actual cash crop can help in soil aggregation. We do that by increasing the photosynthetic ability of a plant. So that's one way. We can also increase aggregation with a soil primer, a bit more expensive and a bit more risky. Otherwise, my favorite is with a cover crop. Cover crops, you just can't beat them in terms of the amount of carbohydrates that pump into the soil. Typically in a really good system with a cover crop, you pump the soil for carbohydrates, which then feed soil biology to then get this feature, but you also reduce the need for a tillage event or a herbicide event. And so typically what that allows is you go from 3 herbicide applications, you have your cover crop over, so the period where you're applying 2. The cover crops will suppress the weeds over what would be your 2 herbicide applications, and you use a final herbicide application to terminate the cover crop and any other weeds. So yes, we're still using herbicide. We're going from 3 to 1, which is a lot better, and we're getting rid of bare ground, and we're pumping the soil full of carbohydrates to do this. So likewise, you could also use tillage in that system as well and effectively disc it in. I'm not too worried about a light disking in the topsoil. It's where you really get tillage to depth, which can be quite damaging. but also exposing bare ground with tillage. They can be quite damaging. But a light disking can be just as harmful as a herbicide, so I think it's muchness. Now, another thing to mention is that it's not just the soil environment that does this or that converts manganese 2 plus into manganese, or the oxidized manganese. It can also be manganese oxidizing bacteria and fungi. So there's some microbes in the soil that will oxidize manganese 2 plus into the unavailable manganese. So what we need is microbes that are manganese reducing bacteria or microbes. Now, my understanding is that all comes with balance. I'm sure there's some of those microbes in worm castings because there's everything in worm castings. But really what we're trying to do is boost the overall biological component of our soil, and that comes with soil aggregation and pumping it full of carbon from our cover crops. So really all of that comes together with increased plant health, increased root exudates, which you're either going to get from increased level of plant health or cover crops, and maybe a soil primer. What you could also do is a seed treatment or a regenerative seed treatment. We've got a video on that, you can go check it out. What that does is put the biology right into the seed, and so it adds that biology and that nutrition right into the seed coat for the plant to use. And so that can be quite beneficial. But I'm not too sure of single applications or single microbe species to apply to your soil. I'm a bit more of a fan of just take a shotgun approach, apply, say, worm castings. You basically get everything in there. You can get a worm casting tea or whatever. But that's a great way to get a lot of diversity into your system. The other thing with soil aggregation is we typically also get an increase in soil organic matter. Now, I think excess soil organic matters reduce the availability of manganese 2 plus, but it also what's called poisons the soil. So to poison the soil, or a soil that has a poisoning ability, is a similar terminology to saying pH buffering. So to resist the change in pH is to buffer. To resist the change in redox is to poise. So soil organic matter and good aggregation poisons the soil, which means you're not going to get this massive swing in manganese availability. It's going to be more stable. So that only comes with good aggregation and soil organic matter levels. And again, that only comes with increased plant health or cover crops or a soil primer. But typically, the approach we take is increase plant health for both your cash crop, or if you're using cover crops, do it for both, to then improve soil aggregation and soil health. Cool, so I hope that really helps with your understanding of managing manganese in a regenerative way and kind of the process that we go through in order to manage manganese and kind of the approach we take, as well as that long-term vision of increasing your soil health to then supply your plants with manganese. If that sounds like something you want to implement on your farm, you can sign up for a free consult where you sit down with me for about half an hour and we'll go over 5 recommendations for your farm. It can include things like this, as well as just general ideas of how can we get your farm using principles like this and everything else in our regenerative management series to then improve your production through regenerative agriculture. Anyways, so if that sounds like you can sign up using the link here, and I look forward to catching up. Thanks again for watching. My name's Teal. Cheers.